Nucleic acids are the biomolecules that make up our DNA and RNA. Nucleic acids are polymers of nucleotides. So one nucleotide is like the monomer or monomeric unit of DNA or nucleic acids, like DNA and RNA. So that monomer is made up out of a nitrogenous base, and it's also linked to this ribose sugar, and then it's linked to a phosphate. And it's got this general architecture of it, the structure. This base, there's many different molecules you can use for this base. We call it a nucleotide base, and these are the letters of the genetic code. These determine what letter you have. Now, many of you know a lot of this from biology already, but this nucleotide with the nucleotide base called uracil is only present in RNA, and this thymine only occurs in DNA, and then the rest of these can occur in DNA or RNA. The main difference between DNA and RNA is whether you have a ribose that is deoxy. So deoxyribose sugar means that you have no OH group here, but in RNA you do. So in RNA it's called ribonucleic acid, and DNA is deoxyribonucleic acid. And this is what we call the 2'. So often you see prime numbers. So this is 1', 2', 3', 4', and 5'. And this 3' and 5' location are actually really important in how these nucleotides link together. Now, one thing about nucleotides is they come in two general shapes. So you've got your single ring here called a pyrimidine, and then you have double rings called purines. And here, I think I have this backwards because I don't know why. It's something that I've always remembered because one of my biochemistry professors told us that if you are a good person, then you have a halo, right? So you're like an angel would have a halo. And so then he said, well, if you're extra good or extra pure, then you have two halos. And so purines are extra pure and they're purely good, so they have two rings or two halos. So these are purines and these are pyrimidines. So I'm sorry about this typo. I think I just keep forgetting to change it. Each nucleotide's bonded to a ribose sugar. Because ribose is missing that hydroxyl group here, these are present in DNA, not RNA. RNA would then have the OH groups here. So if I showed you an example of this nucleic acid where the groups are linked together, so this is now called a nucleic acid made of nucleotides linked together. You have DNA or RNA. There's actually two ways to tell the difference. One is, notice there's presence of thiamine instead of uracil, so that tells me it's DNA. And also, like I said, at the 2' positions, we do not have OH groups. The phosphate groups are hanging off the end here on the left. This is a 5' sugar then, right here. So the 5' tells me about the phosphorus end. So this is the 5' end. Over here is where I would link it down here to another nucleotide. But, I've ran out of room, obviously, but here is technically what we call the 3' end. So the 3' end has to do with the sugar, and the 5' end has to do with the phosphate. And when you're talking about biology and how nucleotides are synthesized, it's important to know the direction that you're talking about. So often in biology, you'll see, 5' end to 3' end, or 3' end to 5' end, and you have to know the differences. We're not going to focus on that here, but it's something that you need to know for biology or biochemistry eventually. Mostly here, we're just talking about structure, understanding structure behind it and chemistry behind it. So here is how one of these strands represents one of these ribbons. Okay, and DNA is a double-stranded molecule, so it actually has a complementary strand that it will hydrogen bond with. This shows you that the ribbon part is thought of as that sugar phosphate backbone right here. And when this twists around into a helix, it allows for the bases to interact with each other. One base from one strand and the base of another strand can hydrogen bond. And these are like the rungs of a ladder. So if you think about it, these go straight across, and almost like a ladder that you can climb up. So staircase, helical staircase. And so those stairs are made out of the hydrogen bonding between the bases of two different strands. And that's what we call double-stranded DNA. We've been talking about the chirality in biology. All the enzymes have a certain shape that helps them recognize molecules to bind to them, and that results in a certain function or action that that enzyme can undergo. Double-stranded DNA, it turns out that there's a special direction of how the DNA is coiled, whether it's clockwise or counterclockwise. This is what we call a right-handed. So it spirals up according to the right-hand rule. So if you stick your thumb up, you curve your fingers around, that's the direction that the DNA is coiled. And you do that with the right hand. So that's why we call that the right-handed rule. But see how this is coiling if you according to your right hand, that your fingers curve toward, let's see, counterclockwise. So you can see if your thumb was pointing up, this is coiling counterclockwise. Anyway, that's the chirality aspect of it. You don't see the other direction spiraling upwards, but researchers, of course, are experimenting with left-handed helices and how they behave. So that's all very interesting research too. I encourage you to look at some of that stuff if you have time or interest. Hydrogen bonding is different between the different bases. So Chargaff in the mid-1900s figured out, looking at the ratio of C, G, A, and T to each other in DNA, and found that these had the same ratio and these had the same ratio present in DNA. And isn't that a cool idea? So basically, you can rationalize that by looking at the hydrogen bonding sites. So cytosine and guanine both have 3 possible H bonding sites with each other, whereas adenine and thymine only have 2 possible hydrogen bonding that they can do. So based on that information, turns out that double-stranded DNA with a higher G and C content have stronger intermolecular forces. So that means they would require more heat to separate them. And just like with proteins, DNA can be denatured by heat, but also you have to separate the strands to replicate the strands, so to create new cells, more DNA. So there's an enzyme called helicase that does that. And so that's one of the important enzymes you learn about in DNA replication. And it's also the basis for a really bad pickup line. And that is, if you see someone that you like and you want to be really witty and nerdy, you can say, well, I wish I was helicase so I could unzip your jeans. And actually, that was a joke that my colleague Sue Nightingale told me once. So anyway, it's sort of embarrassing to say, but I don't recommend you use that line at all. All right, so that's it. I hope that was stuff that you've heard about before in biology class, but that it gives you a little bit more detail about the structure. And of course, this is all stuff you would memorize if you were taking biochemistry class. So I do recommend that you start memorizing that as soon as you can, like all your fatty acids and what they're called, and the nucleic acid structures. I had to memorize guanine, and the 4 bases or the 5 bases, had to memorize the structures of all those. So I encourage you to start making flashcards and doing that if you know you're going to be taking biochem. And that way you can focus more on understanding what's going on in biochem and, learning more stuff like this, the details of how it all works. But for now, I hope this was a good review of the structure.